TV host, and our co-host today is Greg. And yeah, first in, first serve. So this episode is a follow-up from the first episode on psychology, where we covered dualism, which is the idea that the mind is separate from the body. The basics of the brain. Just learning a few basic things that are key to understanding the brain in future episodes. All right, so Greg, the brain, any thoughts?
Not really.
Well, actually, it's very hard to not think. Your brain is always thinking things. If you try and force yourself to stop thinking, it's called meditation. And it's really hard to do, actually. These thoughts, they just keep appearing in your mind, don't they? So I'm sitting here, and then a thought will appear in my mind, but it's not just appeared from nowhere, which is what a lot of people think, I assume. It actually has been crafted in the subconscious, and then you've been let in on it, so to speak.
Right.
Yeah. All right, Greg. So basically humans have the brain is sort of like a layer cake. You can add layers on top of it, and it's really obvious. I think we can all picture a brain. And so the first part of the brain would be a spinal cord. That sort of has its own, that's actually a sort of another central nervous system, because the spine can relay messages without having to consult the brain, which is really odd. For example, Greg, I'll use you in this example. Okay, say if I turn the stove on and I say, "Oh, Greg, just chill, go and stand over there, sit back," and you put your hand on the red hot stove. Do you notice how quickly you take it off?
Yeah, there's a delay, but certainly pretty quick.
Yeah, well, the delay would be even more if so basically when you put your fingers on the hot plate, your nerve, the pain and nerve endings send it electrical, but they don't send it in just one long cable, which would make sense. They send it in little links. So every link has to exchange some sort of chemical, which then will send the electrical impulse up further. So it actually goes quite slow, like meters per second. So the reason why you don't burn your hand as much is because you automatically take your hand off, because the message doesn't have to go all the way to the brain and then all into your arm. It just goes to the spine and then it goes back again. So if we're going to learn about the brain basics, we have to get rid of the second brain, as well, call it, which is basically your spinal cord, because it can control things and it doesn't have to consult the brain whatsoever.
Oh, yeah.
That's why it happens so fast. Before you're aware of it, your hand's already off the stove, isn't it?
Yep.
Yeah. So that's a good one, because people can relate to that. But I guess people must burn their hands a lot. Well, yeah, but it's so this is what I'm getting at with the brain. So basically, your brain controls basically everything that goes on in your body, except for maybe on the DNA cellular level. But if your brain shuts down, all your other organs shut down, because they're all linked. And the brain is the only organ in the body that is protected from things that shouldn't be in your bloodstream from getting into the brain. It's the only organ that you evolved to, that evolution found worthy, or to its advantage for survival, to protect. All it is it just wrapped it up in a fatty layer, a cholesterol, and so only things that are fat soluble can get into the brain. And the brain only wants it's very selective. It doesn't want very many things. It mainly wants glucose, because it doesn't like any other sugars. It has to eat glucose, and it has to also receive the basic oxygen to fuel the energy of the cell, and basically some electrolytes, which are just sort of positive or negatively charged molecules that can cross the blood-brain barrier, as this fatty layer is called. I'll call it the BBB. I'll type it in the chat there. So then it's a good question, is why aren't our other organs encased in this layer? Greg, why did we just pick the brain to protect? What about the liver? That's pretty important. Or the heart? The heart. The heart's actually a weak point.
Yeah, that's true.
Because have you heard of endocarditis?
I've heard of it. I don't remember what it is.
Well, yeah, basically, carditis is the heart. So you have basically your bloodstream doesn't have any natural defenses, your organs. So because most things we consume orally, and orally consumed substances can go through something called first pass metabolism. So they get metabolized, and so they don't. So a lot of this is so they can. This is to protect your body, right? It breaks down things that could be harmful, like ethanol, or hormones that are running around that need to be taken away. Otherwise, you have a panic attack, or something like this. Okay, but the heart is really weak because in hospitals, say they inject you with a medication, that if there's too much bacteria in the water, your brain will be safe because the bacteria can't get across this BBB, this cholesterol fatty layer. But it can get into your heart valves. And how does our body kill foreign organisms? It uses white blood cells or leukocytes.
Right.
And the only place that you don't have these in your bloodstream is in your probably one of your probably one of your most important organs is your heart. So your heart valves don't have any white blood cells. So if you get an infection there, there's not much you can really do except for like have intravenous antibiotics, because the infection there can't actually be battled off naturally, but the brain for some reason has been encased in this fatty layer. Okay, and now I think, Greg, you should maybe, if you have any questions, you can just dive right in. What do you want to know about the brain?
I don't really have any questions yet.
Yeah. Okay. Well, I'll give you a little blueprint. It looks like. Okay. So we'll start at the very back of the neck. That attaches to the of your brain. Sort of. This is where your medulla oblongata is, and this is basically the reptilian part of the brain. So when we were reptiles, we only needed this part of the brain. We needed obviously. So, but we're getting ahead of ourselves. So, just above these primitive parts of the brain, or by primitive I mean they've been there for a very long time.
Right.
Since we were reptiles, actually, we inherited them from reptiles. And then on top of that is a huge, really dense, about size of maybe an apple. It's called the cerebellum. Controls movement, balance, and things like this. And you've got about 100 million brain cells in your cerebellum, I think. But that's not even the fun stuff. The fun stuff is coming up, and it actually gets more and more interesting as you find out how this layer cakes worked. So on top of this, the brainstem and the primitive part of the brain, they put a cerebellum, and then on top, in the next, if you go forward, you've got three layers. You've got on the top of your head is called the parietal lobe. Now parietal, you don't need to worry about what that means. We'll just call it the P lobe. Okay, so the P lobe, what that does is it controls movement, motor skills, and a lot of other things actually. Memory, recalling, lots of stuff, but it's really important because it's it's one of the biggest subdivisions of the brain. Because the brain is just, it's got dozens of subdivisions. Some of them are big. This is a big one. So think of the middle of your head it's broken down into 3 parts. The top part is for what I just said, the parietal lobe. The middle part is called the visual cortex. So the back of your eyes have 2 nerves that run straight to the middle of your brain, and that's where you basically form a model of reality. And it's pretty incredible that the brain, which runs on like a couple of light bulbs worth of energy, can form a pretty reliable model of reality. It's quite astonishing, and we don't know how it works, but we do know that the brain's got some tricks. Because imagine having to, every time you close your eyes and open them again, you have to build the reality all over again. So the brain actually has a little trick to get around this. It's called it uses sort of pre-made shapes, if you want. So basically, if you look at a book, your brain will see it as sort of a rectangle. It knows what a rectangle is like, so it can fill in the rest of the shapes. And if you see something that's like a car wheel, you know it's circular. So your brain doesn't actually have to build everything up from scratch, because it already has these sort of pre-programmed things called geons, which are just like geometric shapes. So it has a basic outline of what it's meant to look at, and only when you focus on it does the detail come out. So if you are looking out the corner of your eye, everything's out of focus. You can only really focus on what's in your right in front of you. Basically, everything else is blurry until you look at it. But you don't have to. The reason when you switch from, say, looking at a pen to, say, a book, and you don't have to, it doesn't take 10 seconds for your brain to build what a book would look like, is because you already have stored away of these shapes, and the shapes determine what model of reality you're going to build. So it's basically literally it's like using circles, horizontal lines, just cup shapes, any shape, and that's basically how the visual cortex works. Below that, you have the temporal lobe, which sort of does auditory analyses, and so that's basically almost all of your brain, the large structures. This is where it gets interesting for me, especially because if you're on the hunt for consciousness, you're probably going to be looking towards the front of the brain a lot, because we're the only species with something called a prefrontal cortex. So in front of the parietal lobe, the POT, okay, so it's an acronym for it. So from the top, you've got the P, in the middle, you've got the occipital lobe, which is your visual cortex. In the bottom, you've got the T, which is the temporal lobe. And then those 3 together control movement and lots of other things. We're finding out that the brain is doing a lot more than we realized. It's specialized in each area, but each area sort of can do things that the other areas can do. Yeah, but the prefrontal cortex, if you asked me what's necessary for consciousness, what's necessary for having a human brain, I would say that the prefrontal cortex is necessary, but it's not sufficient. So you need it for consciousness, but it's not consciousness. So if you didn't have all these other bits that had evolved from the reptiles, which birds and mammals, so you can see how the parietal lobe, which basically half of your brain is devoted towards sort of hand coordination and stuff like this. So the parietal lobe big, and because of that, we have got great dexterity with our fingers. And because of this dexterity, then just imagine the payoff. The whole reason we can build computers and have this conversation is because our parietal lobe got highly the jobs that had to do became specialized, and then because it's sort of the more you use your fingers to pick things, like a chimp trying to do that, they're developing a sort of a situation where they could end up with a sort of much more over generations, of course, much more dexterity. And if it seems that for consciousness, it's very common to have an appendage that can grab and interact with the world. So every single species that is self-aware, it has an appendage that it can use to manipulate the environment. So it seems like if you can manipulate the environment, this encourages consciousness and sentience to arise. Are you still with me, Greg?
Yes, I am.
Cool. If you just hold me up if you need me to adumbrate or clarify anything.
All right, no problem.
Okay, so now but now we're getting to if I can explain how one brain cell works, then we can understand how they all work, because we can just multiply them by 100 billion. So you have 100 billion brain cells. You were born with that many. And each cell is connected to its neighbors with 1 to 10,000 different connections. So one cell could have 10,000 little tentacle feelers going out, which are also, and they meet the tentacle feelers from another cell. So this is, and then if you link enough of them up, this is how you get like a pathway going, like the dopamine pathway through the nucleus accumbens pathway. I don't know if you know about any neurochemicals yet, but that's not important. We'll get to the main ones in a sec. So serotonin is very important. Do you know what serotonin is?
That is the.
Well,
oh, go ahead.
No, please.
I can't remember them all. Serotonin and melatonin are opposites, basically.
They look very similar, and I'm actually kind of surprised that we haven't found serotonin in, say, like trees.
Yeah.
You know, molecules that are like 1 or 2 atoms different everywhere. DMT especially. Dimethyltryptamine is found in so many different plant species, and it's that and the magic mushrooms ingredient thing that makes you actually trip, called psilocin. That is 2 atoms different from serotonin, and serotonin is your main man. It modulates your sex drive, your hunger drive, your sleep, how content you are, how happy you are. So that's why lots of medications target serotonin, because if you can control serotonin, a lot of the things that people think of consciousness are actually modulated and moderated by serotonin. And it seems that there's a lot of interesting research, and I bet you there's a lot of Nobel Prizes waiting for someone who can figure out how this serotonin and dopamine, adrenaline sort of network in the prefrontal cortex, how that all fits in. Because if I had to put in the brain, that's where I'd sort of point. But it's starting to look like consciousness isn't hiding behind our eyes. Like, you know, if you look out, it feels like you're looking out through your eyes. But that's not really what's happening. Do you follow me, Greg?
Sort of.
All right, so the light's coming in and it's hitting your eyes. It hits the back of your eye. You've got a little area called a focal point or something. And so basically that's why the rest of your vision is blurry unless the thing you're looking at, because unless the light hits the focal point, that's the only part that isn't blurry. So the rest can be blurry because it's not important. It's not what you're looking at. If everything was clear and like that, your lens, and also it wouldn't be possible because your lens wouldn't be able to do that. It wouldn't be able to make a perfect image everywhere. That's why we have to look around. But okay, so the light hits the back of the retina, and you only have 3 different colors ways to read colors. So you can see blue, you can see red, and you can see what's the other primary color? I think it's yellow, isn't it? Yeah, because you can make green with green. You can make green with yellow and red, though, can't you?
Yeah.
Yellow and blue. Yeah, so yeah, it's not green. It's yellow, red, and blue. So so basically, it's like a camera. Your eye is like a camera. It's when the light goes through these receptor cells for light, which are actually the cables that connect them to the retina, are actually in front of the receptor cells, which is completely backwards. You would think that it should, because this is evolution. It does things in a very odd way. So basically, that light comes in, hits the retina, but the light sensitive cells are actually behind a cable. A cable is in between them, if you know what I mean. So it's just one of these things that evolution did, because it was basically just took the simple way out. It could have rewired everything, but it didn't rewire everything, because that's not how evolution works. What it does, it just basically uses what it has and adds stuff to it. So it already had a light sensitive cell there, so we're not going to move that around. We're just going to make the cables go in front. But anyway, once it hits one of these 3 things, your brain can make up to 1 to 10 million different colors, and you can actually distinguish between these colors, 1 million different colors. So it is like a camera in that sense. But then you've got the nerve that connects your eye to your occipital lobe, your visual cortex. So, but when the message gets sent back through down to what's there, and and so basically you're constructing a model of reality. There's no reason to think that the world actually looks like this. I mean, because it certainly doesn't look like this to bats or to you know what I mean, to other animals. But right, we think the world actually looks like this. But it is true to say that in my profile pic that is blue. And by blue, I don't mean the color that blue. I mean the wavelength of light blue. So somehow your brain is just, it can manufacture a complete cosmos inside your head.
Right.
So,
like red and blue are magenta, or what we see.
Yeah.
But they aren't. They're just red and blue.
Yeah. Well, we can only see red, blue, and yellow. We can't see any other colors until we've mixed them together and made them in our head. So these colors, our eyes don't only perceive 3 colors, but our brains can sort of, like a computer, like a simulation. And so basically that's, and the same is true with your hearing. So basically the brain collects information from the external world in order to help you survive better and pass on your genes, which is the name of the game. If you believe in evolution, which is, you don't really have a choice but to believe. Okay, so the last thing I want to say on this is that is a cell, what happens in a single brain cell, it'll get an electrical signal from a cell that's linked to it, and just one cell back, okay? And so once it receives this electrical current, it depolarizes. It's like turning a switch on and off. It actually goes from I think it's minus 7 millivolts. So it's in the negative. Your brain's actually in the negative to start with. And then once the electrical signal hits it, it flips its polarity. So it goes from positive, goes up to like plus 55 millivolts. And then it takes a while for it to go back down. That's called the refractory period. And the obvious analogy here is when you have an orgasm, you can't just have another one straight away. You have to wait. That's exactly what happens in the brain. It actually, because once your brain switched the polarities, it actually has to, usually the sodium potassium pump to get all of the positive ions out of the inside of the cell. And so it's basically got a gradient that's ready to go off. But it either goes off or it doesn't go off, which would be, if this was all there was to the brain, it would be so easy to work out, because this is like a computer, it's binary. So you either have this electrical polarization, this is called an action potential, and it either happens or it doesn't happen at all. So there's either enough electrical signaling in to actually fire it, or it doesn't fire. But if it does fire, then this is 60-year-old research. 60 years ago, we didn't know that serotonin existed, let alone that it's the most important understand about the consciousness and the mind and the things when we want to learn about the brain, creativity, intellectual abilities, this sort of stuff. So, okay, so the cell has fired, it's done the action potential, so it'll send an electric current actually down the cell and down the dendrites, and it can have thousands of these dendrites, and then these dendrites are what connect it, one cell to another. So if let's follow the dendrite all the way to the end, and if you see at the end there's actually a physical gap in space between it and the receiving end of the dendrite. So you got a presynaptic dendrite and a postsynaptic dendrite. That's basically the part where the electrical signals hit, and the part where the postsynaptic dendrite is waiting. I think is waiting for some information. It's got a protein or something curled up in there, and however that protein, this is actually as far as we can go. We don't know what happens after this, but what happens is the electricity hits the presynaptic the presynapse, and there's these little bags. They actually are little circular balls, and if you pop them open, they're full of neurotransmitters. They're full of if it's a dopamine receptor, it's full of dopamine. But we'll be talking about serotonin here. So there's lots of these little vesicles, little balls filled with thousands and thousands of serotonin molecules. And so when the action potential goes off, your vesicles, they can pop open and they can release the serotonin into the synapse, which is a physical gap. It's just floating around in the middle of your brain. It's a physical gap in space. So all 100 trillion of these connections, they're not actually connected. Every single cell has a physical gap between the other ones. But this gap is important because the serotonin molecules will float across, and they this is why it gets so complicated, because it's not binary anymore. It's not the action potential either happens or doesn't happen. What happens with serotonin is it can land in the receptor, but it has to enough has to land in enough receptors for to the proteins. So most serotonin receptors are called G protein coupled receptors. That's just fancy for saying there's a protein that's attached to the receptor. And these proteins are huge because proteins are made of amino acids, and a molecule's like about 10 atoms, or if you include hydrogen, it's more. But we're talking it's like 1,000 times as smaller. We can't read serotonin, but what happens when the serotonin hits the receptor is what it's all about. That's what we need to do more research on, because what happens when a molecule hits a protein is the protein changes shape. It's kind of interesting. It's called conformation. And so basically what the protein does, it actually reads the shape. I use read in quote marks. It reads the shape of the serotonin molecule. And after it's read the shape, it actually bends and twists and contorts into a different shape. And that change in shape, it alters the enzyme release protocols for that cell. Okay, so say if a lot of serotonin hits it, and this protein conforms, and then it releases these enzymes in a special way, and then the net result of that is that your next neuron in this pathway will release more serotonin. So, and whenever you release increased synaptic serotonin, you feel happy. You can't help but feel happy. You can't help but feel content. You can't help but feel because it's basically a happy molecule. Like it's the me molecule, if you want. Whereas dopamine is the greedy molecule. I sort of use the analogy of it being like the crack dealer on the street. Right? So we evolved to eat the tastiest berries, right? But
Right.
once you get these dopamine, sorry, Greg.
Okay.
All right. So yeah, dopamine is another main player in this prefrontal cortex. It's what happens is dopamine controls pleasure. So literally the only reason a hamburger or whatever you like tastes good is because of dopamine gets released. When dopamine gets released, your brain goes, "Oh, like that, I want more of that." So this is how animals survive. They eat something that's high in fat or high. It tastes good, and your brain rewards you for doing that behavior by giving you dopamine. You know what I mean? It's giving itself dopamine, but and this is how your brain can trick itself into addictions of any sort, really, because you evolved to eat the tasty berry than to get the dopamine. But so suddenly you're thrust into the 21st century, and there's all these artificial ways to increase your dopamine. So basically, so if you're an alcoholic and you drink alcohol, that releases dopamine. So you have a sip of beer, and your brain goes, "Oh, that was good. I'll give you here's some dopamine. Yeah, that's nice. Okay, but now I want that was pretty good. I think I want more dopamine." So you drink more, and then this is how it gets out of control, because a circle. Your brain will say, "Look, let's get some more dopamine," and the only way to get the dopamine that we want, the tasty berry, is by cracking another beer or whatever it is, or having another cigarette, another line of whatever is popular in the US. Assume it from there. Yeah. So basically, your brain. This is what happens to the minds of addicts, because this is what I studied actually, with addiction studies and psychology. So an addict their brain has is run out of dopamine, and by run out I mean it doesn't have enough to be functional, because dopamine also is one of the molecules that gets you out of bed in the morning. It controls motivation, in other words. So if you have messed up your dopamine pathways, you basically have to wait for your brain to renormalize, or you have to artificially alter the dopamine levels. And if you do that, you think the best way to do that would just be to eat a whole bunch of dopamine, right?
Oh no, no. Wouldn't work that way.
Well, you're right, it doesn't work, but it's interesting because if this comes back to BBB, the blood-brain barrier, dopamine actually can't cross it. It has to be made inside the brain. So it's kind of frustrating, because it would be a really good, and it would be a natural way too, because it's pretty much harmless. It's just doing what the brain does naturally, but you can't get it into the brain unless you alter it. So you can alter it and make it into, say, a really close chemical structurally, like amphetamines or L-dopamine, cathinones. They look like dopamine, and the reason that they feel really good when people do them is because they're releasing dopamine, but they're also forcing it to be in the synapse. So they burst all those little vesicles up, those little precious little bundles that you've stored up. It comes in there and just bursts them all open. And then it says, no, because the dopamine doesn't want to stay in the synapse and keep firing the cell over and over and over again, because it gets tired, it gets worn out. These action potentials, this is why people who use say meth for a decade, every day for a decade, they literally can't do anything unless you give them amphetamines, because they actually can't. Their brain, they've seriously damaged their dopaminergic networks, and they've also not so as dramatic, but they've damaged their adrenaline networks, because it's just the dopamine's floating around in the synapse, but there's these little, think of them as like little garbage trucks. These enzymes come and grab the molecules of dopamine. They actually try and recycle them by putting them back into the presynaptic cleft. So they try and put them back into those little bowls, into the vesicles. But these things, the drugs that prevent this from happening are called, so they prevent the reuptake of dopamine. So they stop the enzyme from working. So first of all, they push all the dopamine out as much as they can, and then they stop it from leaving. And then they also, the molecule itself can fit the receptor of a dopamine receptor. And then why the hell can a molecule that's not meant to be anywhere in the brain, how can that fit a receptor? More than that, it fits the receptor, and it actually conforms, and just like it would if it reads it, the protein reads it, and it's never seen it before, it never come in contact with it. And this is why people get, this is why drugs actually change consciousness, because the proteins try and read the new molecule, but it's not what they're used to. It's not dopamine. It's not adrenaline. It's not serotonin. So they go, okay, the protein bends or conforms into a new shape, and so you can imagine the implications of that. I mean, the protocols for releasing enzymes can get switched up so dramatically that people can sort of, I don't want to make it sound too much like a panacea, but you can sort of upload new software in a few hours if you do it correctly. There's been studies where they've improved the success rate for quitting smoking up from 10% to 90%, which is a pretty which is pretty impressive, because smoking kills 50% of people who do it. But if you take this study showed that if they only one person went back to smoking, that's why it's 90%, not 100%, and they were just a light smoker after that. So it basically, but this drug is a Schedule 1 drug in the USA, so you can't actually, it's so hard to get research grants. And this is the tragedy, because if you want to understand the brain, you have to understand serotonin, you have to understand dopamine and adrenaline, and you have to use probes to get in there and learn what's going on. So for example, I want to find out where psilocin, the magic mushroom drug, goes when it enters your brain, because this is actually how they discovered serotonin. They did it with LSD. I'll use LSD. So LSD, it was invented just before they discovered serotonin in the brain, and they wouldn't have just even known to look for serotonin in the brain, because they didn't know what was controlling consciousness. Half the people thought, oh no, it's just an electrical thing. It's going to be easy. It's just on and off with the electricity things. They didn't know about synapses. They didn't know, because they wondered, how can it happen so fast? Because electricity obviously travels very fast, but a chemical reaction takes a lot longer. But they weren't really looking for a neurotransmitter that modulates most of the things we think of when we think of sentience. So but a few clever people put two and two together. They noticed that LSD affects basically all the things that consciousness hinges upon when we define it: our mood, our perception, our hunger, sex drive, you name it, and it's there. It's modulating it. So if you can somehow manipulate this most important neuro receptor and upload some new software, because the proteins, this is pure chance, they change into shapes that they can't do unless LSD is put in there. And also they'd said, wait, but hold on, that's not enough for us to start looking for serotonin in the brain. They knew serotonin existed. They just thought it was a neurochemical for digestion and things like this in rats. But they didn't realize how important it was for the brain. And so this is 1950s, okay? And so then some smart person looked and said, wait, hold on. There's actually a molecule of serotonin embedded inside the LSD molecule. And so they said, wait, it affects your consciousness and it's got serotonin in it. If serotonin is in the brain, surely serotonin controls a lot of these functions that we're trying to pin down with consciousness. So basically, this is you could sort of plant a flag and say this is the start of modern psychology, neuroscience, basically. Before that, neuroscience was a pretty empty field. Isn't that weird?
Oh, yes.
How LSD actually directly led to the discovery of serotonin. And ever since then, we've been trying to figure out how to make people how to make people's brains into things that make people into people they want to be. And how do you do that? You change the wiring in their brains, and you can do it naturally too. You can do it with exercise, diet, sunlight. All these things boost your serotonin. But the thing is, if you do it with an artificial substance, you can bend the proteins into ways that it's not meant to be, or that it has never been bent before. So the way it releases its enzymes will be changed up radically from what it would be if it was just reading serotonin. I hope I'm not getting too jargon and technical.
No. I just don't understand how it's. I think of it like a key that fits a lock, and it's making its own special key that doesn't fit a natural lock or something. It doesn't seem right.
No, dude. Actually, what you were saying is actually what people believed all the way. People believed it probably right up into the 70s, because they didn't realize that these proteins, can't just be on or off. So basically they release acid enzymes, and I think one of them is called arachidonic acid, for example. But the order in which it releases them is very important, because there's multiple ways in which you can switch it up. So so we don't actually know how far these things can take you, but we know there's evidence now to show that when you take serotonin's cousin psilocin, parts of your brain, two neurons that haven't connected for years or ever, they start to reach out their little dendrite feelers, like two trees reaching branches out across towards each other. And this happens in real time. If you had if you had a microscope, you could watch your brain, the growth happen in actual time. So it's basically, think of your brain as the hardware, and the subconscious and your conscious, that's sort of software humming along on your hardware. But your hardware and your software it's like a computer is a good analogy, because a computer has the hardware, but the software has this to run on, and that's the hardware. So your mind is running on the hardware of the brain, but we can't actually, until we actually figure out we don't know what happens after these proteins unwind and release enzymes. And because we're finding out that you can do it give you psilocin, it'll be a very different experience than if I give you almost an identical molecule like DMT. They're similar. They're both serotonin 2A agonists. Okay, so basically they go to serotonin, and you got subgroups. They all are serotonin receptors. They just moderate different things. Okay, so if DMT hits the receptor, it'll do one thing. If serotonin hits it, it'll do what it normally does. And there's literally millions and millions and millions and millions of unique combinations. And so it's not like a key opening a lock, which is what we used to think. That would be really good if it was like that, because then it would be easy to understand, and we would have probably solved the brain by now. But the reason it's so complicated is because this proteins can change shapes dramatically, and when they change shapes, they change, and that means that they change the way that the cell behaves. Yeah, so basically that's how. And so what happens after these enzymes and the G protein? Well, we don't know, because 2012 Nobel Prize in chemistry was for research on G proteins. It's pretty new stuff. It's new to me as well.
Yes, very.
Yeah, but so with the lock and key model, like where serotonin is the key and the receptor is the lock, that is accurate. But think of it more like this. So you can have two types of key. Then there's like a baseball glove. You can throw a ping pong ball into it, and a ping pong ball is gonna hit the receptor, but it's gonna bounce high dissociation rate. So it's got a high affinity. It fits the receptor perfectly, but it doesn't stay there, because it dissociates away and gets recycled. Whereas, say if you throw a sticky ball and then it hits the mitt, that's got high affinity because it fits the receptor, but it's also got a slow dissociation rate. So it sits on the receptor for ages, and then that causes the cell to respond by firing. And so the slower the dissociation rate, the longer the effect, whatever it is, will be, whether it's serotonin from gambling or whatever, or if it's a drug causing it. Right. Yeah, but if they do these studies, you can't map out where serotonin 2A is in the prefrontal cortex, because you need to do the mapping. They're Schedule 1, so it's easier to do a study with cocaine than it is with these things, and they're non-toxic, which is actually, so they basically mimic what the brain does. The toxic drugs are the ones that violate the brain's natural processes. So I think that's basically the brain in a nutshell, brain basics. But yeah, I guess we can just have a little chat for a bit longer, maybe for another 10 minutes. Yeah, so Greg, what do you find? I think that, are you just like bamboozled by the whole idea of consciousness?
Oh yeah, it's
good. I certainly am. I think it's one of the biggest mysteries up there with like pre-Big Bang stuff, like what caused the universe. I think we're going to solve that one first by the looks of it.
Yeah, probably.
It looks like it came from nothing, but I'm not going to talk too much on that because that's not my area of expertise.
Nothing isn't really.
Yeah, it's still fascinating though. Yeah, exactly. Empty space weighs something. So if empty space weighs something, it has potential energy, and that potential energy could cause the universe to. And the cool thing is you can imagine a whole universe inside your brain, and your brain is so complicated. So basically let's just assume we've got how a cell works now, and we pretty much do. We understand how. We're getting close to there's no more layers of onions, right? It's from the onion. But the problem is, even if we figure out how a cell works, we still have 100 trillion synapses to map out, and that's more than there are suns in galaxies.
True.
By a lot. So what, you get 100 billion neurons. There's about 100 billion stars in the average galaxy. So it's basically like your own little universe in your head.
Yeah, you can look at it that way.
Yeah, and also we I guess you could call it odd in another way, in that in one of Carl Sagan's books, he was trying to figure out this intelligence, and so a good way of measuring intelligence actually is how big an organism's brain is compared to their body size. So elephants have big ass bodies, but they also have big brains. But if you look at the ratio, it's actually much lower than two. The winners are humans and dolphins. But dolphins are kind of stuck. They're not like us. They got flippers. We have hands. So all the other organisms that have conscious, they can see themselves in a mirror, say, and there's only half a dozen species that can do this. Dogs can't do it. Elephants can't do it. But crows can do it, and octopi can do it. And I think octopi can do it maybe because they have tentacles. So maybe you need an appendage to interact with the environment to get some sort of sentience going. And crows obviously use their beaks for things. But if you're in the water, you're a dolphin, you just catch fish all day and you play. So you're not developing past that. You never, unless you have some way to manipulate your environment, like the octopus or like a monkeys and apes who also have this skill, and I'll include us in the apes part. Or what do we all have in common? We have a way to manipulate our environment. And yeah, but this gives me hope for intelligent life elsewhere, because the common ancestor between humans and octopi was 600 million years ago. So we did not evolve. We didn't just inherit consciousness from that. It evolved separately multiple times.
Yeah.
Yeah. So brains, intelligent, sentient brains, seem to be actually an evolutionary beneficial thing, because several species have it, and all the ones that do have it are doing quite well, except for the ones that we've screwed over, like gorillas and stuff like that. But if it wasn't for humans, they'd all be doing okay.
Maybe they would have been better.
No. More what?
More advanced.
Well, yeah, we'll see. It's tricky because advanced. We're defining what advanced is, and then we're seeing if they fall short or not. So it's anthropocentric. It's human centered.
True.
It's because we can't help it. Hold on, I got to go, man. I'll thanks for listening through FSTV. Thanks to Greg. Bye.